Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Max, and I'll be your host. We are delighted to have our wonderful sponsor, Dyson, with us today, who'll be presenting this webinar titled Defense in Depth, Best Practices for Contamination Prevention. Today's speakers are David Grote, VP of Strategy for Grey Matter Partners and a Principal Consultant for Canadian Group, and Sarah Finnerty, America's Marketing Manager for Dyson. Now I'll provide some housekeeping information before we start. First of all, I'd like to welcome you to our webinar platform, ON24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask questions to our speakers. You can do that via the questions widget. Just type your question in this box and click submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. If you require any assistance, please use the help widget, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome David. Over to you.
Thanks, Max. Hello, everyone. I'm David Grote. Throughout my over 25 years in biopharmaceuticals, I've been leading quality and operations groups at companies like Amgen, Teva, and Immunocore. I've seen how contamination challenges can impact patients and the company's bottom line. In my role, I'm driven by leveraging innovation to solve issues, ensuring we achieve those highest levels of quality and regulatory compliance as cost effectively as possible. We owe it to the patients that we serve. I'm really excited today to present my perspective on this critical subject, contamination, which I call Defense in Depth, Best Practices for Contamination Prevention. This presentation is going to be a high-level overview and summary of critical tips. If you'd like to explore any of these topics in a deeper, focused conversation, please reach out after the session. While regulatory compliance is, of course, foundational, I'm not focused on that. I'm more focused on the strategic application of principles rather than reviewing the regulations or covering details on cleanroom classifications and things of that nature. To maximize our time, I'll focus on broad strategic principles and rather than diving into those types of details. And these principles really apply across all operations wherever contamination will be a concern. By contamination, I'm referring to all types of contamination: microbial, viral, chemical, particulate, cat hair, you name it. Anything that gets into your product or process stream that shouldn't be there, I'm considering contamination. And these principles that I'll go through really apply to controlling and preventing all of them. At the end, I hope you'll stick around because as I end, we'll wrap up with a fun challenge, what I call Spot the Problem game, where you can test your eye for common contamination risks and hazards in a real-world scenarios, but basically photos from facilities or simulated facilities around the world where problems that I've seen throughout my career. So we'll go next. Going to kick off with a story that many of you may remember. It was a number of years ago now. And this was a Genzyme facility in Austin, in Massachusetts. And they had a serious issue where they started losing their bioreactors. The bioreactors just started dying. And what the investigation determined was it was a viral contaminant. This is a MMV contamination, which is mouse minute virus. And by that name, you probably can guess it's a pretty small virus. It's a 20 nanometers in size. So 5 of these viruses could fit side by side and pass through a 0.1 micron sterilizing grade filter. So if our filters, viruses are, our viruses have no visibility to our filters. Our filters don't necessarily control them at all. For additional perspective, 15 of these viruses could fit side by side through the pore size of a HEPA filter, which are usually 0.3 micron. This was a real wake-up call, particularly for the biopharmaceutical industry, and it resulted in a consent decree with Genzyme. They had to pay the federal government $175 million in profits. The total cost of this is significantly higher than that, because there was also a loss in public trust and reputation, and they had to move multiple products away from this facility. A more recent example of contamination and the impacts many of us have been talking about across the industry is Novo's recent issues with their Bloomington, Indiana facility. You may have noticed last month they got an official action indicated letter from the FDA, and this was due to systemic failures in contamination control, among other things. They found at the site there were repeated discovery of extrinsic contaminants, including cat hair, human hair, and pests in classified areas and in product components. And so this was an unacceptable risk, clearly, to patient safety and product quality. And they recognized also that the quality systems in place there had not caught or adequately investigated or corrected these issues. And this is the latest in kind of a string of significant 483 and other observations for this facility. So this has had quite a dramatic impact on their business, as well as the several companies that use them for fill finish operations. And the lesson from this really is that contamination control is a very important and critical part of your operation. And you need to have a robust controls and a strong quality culture to prevent the impact that it can have on your business. This facility was part of a recent purchase by Novo of Catalent, and it was the total purchase for the 3 fill finish sites, that one of which is the one talked about here, was $11 billion. So huge money invested in this facility, and obviously a huge value for Novo, and now that is being placed into jeopardy because of this issue. So that's a cautionary tale. Now, sometimes when people read these things, they think, oh, it's too bad for them, but that's not the right take-home message. If you are involved with aseptic filling or any type of clean room operations, the real take-home message is, I better make sure this isn't us, and take a good close look at your facilities and controls and make sure that that you don't have these same issues, because the FDA will be focused on these, and other regulatory agencies will be focused on these. I've seen it many times over my career, that small observations that are isolated observations at various facilities around the world can lead to kind of a snowball effect, where investigators are saying, oh, I better make sure that when I do my next inspection, this facility doesn't have the same things that I saw at that facility. And so the bar is being raised, and it's time that we recognize that and adjust. So you all are here, so obviously you feel that contamination control matters, but it is a huge issue, and it's still, I think, rather misunderstood by many in the industry. There have been a lot of lapses, and I just talked about the regulatory issues, but more significantly are the many cases of deaths, injury, and patient impact from this. If you follow me on LinkedIn, you've seen a number of my stories. I post quite frequently about these things. Unfortunately, I have many years of experience with it. I have a lot more stories coming, so I do encourage you to follow me and check on that. Check on my feed as I share these stories. And it's been fun for me in my consulting role to be able to share these stories and that I've experienced over my career. So patient safety, product integrity, microbial contamination can have many effects, not just the microbes themselves, but they also can impact the product. And some of those ways that they impact the product might not be detectable by our routine assays. Particulate contamination, like cat hair, obviously is a huge issue. And laboratory contamination can really spell disaster. Next slide. So I'm not going to spend a lot of time on regulations, but I would like to mention the recent revision to ICH Q9 and also the contamination control strategy, which is defined by the EMA and Annex 1 revisions. So the ICH Q9 is a policy around proactive quality risk management and the expectation that you have this in place. And for contamination, the way to approach this is to have a robust contamination control strategy. So it's a holistic approach. It's science-based, and it is comprehensive and based on evidence. It's also a living document, so it needs to be reviewed continuously and improved and monitored as your process changes, your facility changes. And as you learn things, you should revisit that document. The ultimate goal for this is to safeguard product safety, patient safety and product quality. If you are focused on patient safety and product quality, you will do well in inspections. That's really the take-home message. The inspectors aren't there to collect observations. They're there because they've been tasked with ensuring that we in the pharmaceutical industry are producing safe, effective products that meet their criteria. So one of my favorite analogies for this is to think about the contamination control as multiple layers of control. Defense in depth, that's what that's referring to, is the process is at the center of many different rings of defense and or layers, if you will. Not layers like cakes or parfaits, but layers more like an onion, with each layer of control helping to protect the innermost part of the process, which is the product which ultimately goes into the patient. So when you're talking about manufacturing, it's always a good idea to back up and think about the big picture. What are the elements that go into a manufacturing process? And these are usually thought of as the 5 M's, manpower, machine, materials, methods, measurement, and then this bonus one, which is Mother Nature or the environment. This is more the external environment. The internal environment, your clean rooms and things like that, would fall under machine in my model here. This is just a way to think about things to make sure that you're looking at all aspects of the control. To speak briefly about Mother Nature, that last element, it can have a huge impact. Seasonal variation, for instance. I've seen problems with bird migrations, bird droppings, and in the parking lot, changing the microbial load inside a facility. Cold snaps that drive pests in. I've seen that with rodent infestation over the years. Humidity and temperature changes can have a big impact on your facility or your product if it's in transit. I'll be briefly touching on each one of these 5 M's in this presentation, just as we go through. So taking a look at a slice of that onion, if you will, these are the layers of control I'm talking about. This may not be all inclusive, but at the top are the sources of contamination. So this could be animals, it could be pests, it could be bacteria on your skin from people, all of those types of sources. And then these are all the various layers of control that you have. You have your site and facility design controls, your HVAC and environmental controls, the personnel and gowning controls. You have control around materials as they come in, not just sanitization, but also quality inspection, ensuring that some testing and things like that, but then sanitizing those materials or sterilizing them before they come into your process. You're using closed process equipment. You have sterilization steps in your process, in process and product testing, and then finally the patient. It's very important to think about this as multiple layers of defense. Not a single one of these layers is foolproof. Things can get through these layers, many of them. But together they provide these layers of protection. And if you have a deviation or an issue, like I shared recently an example of a leak on a disposable bag, if that leak because it happened in a clean room, helps to give you some assurance that that the product will still be okay. And the fact that it's in a clean room, it's gowned operators are working with it, all those things, and you have other controls in place. It's these layers that give you that protection. Because not any one thing can control all the possible hazards. Another very important point is that you cannot test quality into the product. There's no way that you can do all of the testing that you would need to do on release of a product, particularly a complex product like a biologic, and be able to find anything that could be wrong with that product. We rely on the process controls in place, and I'll give you an example of that a little later in the presentation. So a key part of this concept and of your contamination control strategy is the proactive risk assessment. And there's many tools that you can use for a risk assessment. FMEA is one of the classics. It's not the only tool. There's other tools. Some people don't like FMEA. I still think it's a useful tool. It just needs to be used with caution. And I have some caveats on how to use it in the future. But it all starts with process understanding. That's the base of doing this effectively. And that means you need to be able to understand all aspects of the process. And to do that well, you really need a diverse team, not diverse in terms of race and gender and things like that, but diverse in terms of perspective and diverse in terms of training and background. If your room is filled with just chemical engineers and they're talking about these things, they may miss the microbial risk present, and vice versa. If just a bunch of biologists there, they may miss issues that an engineer, somebody with a physics background, would be able to pick up. So you need a diverse team. Failure modes effectiveness is one way of helping you to prioritize risks and understand where things need to be improved. And then that output can be either used to redesign the processes or controls or improve your detectability around issues that you may find. So some of the caveats. So just like any tool, failure modes effective analysis is a tool and it has its limitations. It's a prioritization tool that should be thought of as that. It's not a solution. As I mentioned, it's not the only tool. There's multiple ones listed in ICH Q9, but this one is first, I think, because it's still very popular. You need to take action based off of your FMEA. The success relies on the team expertise, and so you want to involve a diverse team, including operators, suppliers, and key experts across different disciplines to capture that knowledge. You want to do this rigorously, especially for something as important as contamination control. The expectation is that this is a rigorous exercise and it's well documented. It's also important to not confuse failure modes with root causes. Failure modes is best think about them as a mechanism. What's the mechanism of something bad happening, not the actual thing that's happening. That's a key distinction. You want to integrate those findings into a control plan and think about mechanisms, as I mentioned, and then understanding those mechanisms and doing what you can to redesign the process or tweak things, improve things so that it can reduce the risk of that happening. This is part of the contamination control strategy. Now, I say mandatory. It doesn't not mandatory that you do an FMEA, but risk assessment is a key part of this, and FMEA is one tool that you can use for that. So I'm not going to go into great detail on this, but this is an example of what a typical FMEA sheet looks like. A lot of times these are set up in Excel. The team approach is critical. You want to map the process, identify those failure modes. For each, you're looking at three aspects. First is a severity score, and this could be from no impact to ranging from no impact at all to loss of batch to patient illness or death at the most severe. And occurrence is from extremely rare, meaning that after hundreds of batches, you think this probably still wouldn't happen. Or extremely common, meaning that this is guaranteed to happen, a good chunk of the time, a good amount of the time. And the detectability score, confuses people a little bit sometimes. It's not just, can you see a problem? Do you have the right analytical tools or detection mechanisms to see that problem? It also can be that you have controls in place so that that problem just could not happen. The critical thing here is that the existing controls or detection mechanisms that you have will either see the problem before it has an impact or prevent it from happening entirely. And then the way this tool works is you multiply those three numbers together and you get a RPN score. Some people use a ranking system or something like that, but there's no real rules on this except for do what makes sense for you and your team. And this can help you assess relative risk and improve control. And obviously you want to document your work, as we have to do in anything in the GXP environment. So one of the examples of this and how this could work is for a process step. And this is a typical biopharmaceutical process step where you make up a batch of media, sometimes in an open container like these operators are doing in this photo, and then you sterile filter that through a sterilizing grade filtration. I don't usually claim that that step is sterilizing, but it has to be really practically sterilizing, or else the bioreactor will come down contaminated. So that's typically a 0.1 micron filter. And in this example, we have growth media that's prepared warm because they have to get some things in solution. And so they dump powdered media components into an open tank, and then they hold it warm. And then they mix it, and they can hold it up to 12 hours before sterile filtering. I hope that some of you are thinking right now, ooh, that doesn't sound very good. These are the questions to think about. What are the risk mechanisms? What's the severity, occurrence, and detectability of each? And then how could these issues be mitigated or improved? So I shared an example as one of my top hits, I suppose, on LinkedIn, where I shared it was called "The Bioreactor Died," and so far that one post has been seen by, I think, 90,000 people across the industry, and so it's got quite a bit of attention. In this incident, we had a bioreactor that crashed all of a sudden. All the cells died. We thought, oh, it's contamination. Looked in the culture. There was no sign of any bacteria in there at all. The mammalian cells were all dead, and the classical dissolved oxygen and pH changes had happened, but there was no bacteria present, and so we couldn't figure it out. And we ultimately investigated, and we determined that it was Bacillus cereus, a top culprit in biopharmaceutical contaminations. It's also the root cause of food poisoning a lot of times, particularly with rice dishes. And what happened was that this Bacillus cereus bacteria had grown and bloomed in the culture, the media, before it was sterile filtered. And the sterilizing grade filter removed those bacteria, but Bacillus cereus can produce exotoxins, and like any bacteria, they produce proteases, they produce other things, other byproducts, as well as their own DNA, all of those things. In this case, the exotoxins that the Bacillus cereus had produced passed right through the filter, as they will, and they killed the cell batch. So this was a real wake-up call for me and for many of us, because this was just a lost batch. But had the same issue happen downstream, further downstream, like preparation of a final formulation buffer, the buffer that actually goes into a pre-filled syringe or a vial, people could die from this. And so it was quite a wake-up call. Some of the lessons learned really are that filtration can hide problems. Everywhere you have a sterilizing grade filter, it removes the microbes, but it does not remove the other things that may be in there that the microbes may have left behind. We also don't have any testing for those things. There's no assay for bacterial exotoxin that covers all of them. We have assays for endotoxin, nothing for exotoxin that is comprehensive, and that you can use on a routine basis. So you need to have bioburden control throughout the entire process, especially before filtration. And risk assessments, as well as sampling and monitoring for bioburden everywhere in your process upstream of a sterilizing grade filter is so key. Those are some of the lessons learned from that one. So moving on to design of facilities. So it's very important to have a effective layout for which controls airflow, personnel movement, material movement, and all of those things can help prevent contamination. When you're designing clean rooms, you want to make sure that the surface finishes, the corners, and the drains are covered and are properly designed to facilitate cleanability and sanitization. Unidirectional airflow and pressure differentials are essential design elements, and these will vary a little bit depending on your process. Sometimes, if you're using materials which can be hazardous to people, you may have to have controls both ways. So your operators are protected as well as your operators are protected from the materials as well as the materials are protected from the operators. So it can get quite complicated, and different clean rooms have different design elements. I'm not going to go into all of that. You can take a couple of days course or more on each of those topics. But that facility design is really key. An important point is you can take an existing shell building, just an open space, and build clean facilities inside this. When I was with Teva, I did this in a facility in Westchester, Pennsylvania. We took a building that was had been a warehouse before, and it was just a vast open space. And then we built a really nice state-of-the-art clean room facility inside it for cell banking, cell culture, and aseptic fill finish for vials and syringes, and in all under one roof. And so it's possible, and the facility it runs terrifically. But it's very difficult to take an existing interior and try to convert it to clean space. I would recommend that you don't do that unless it was already designed with at least some of those elements in mind. It's probably cheaper just to tear it out or find a different place to build. So particulate control in clean rooms is a key layer for defense. And so I'm going to say a word about our sponsor here, Dicem. So I first discovered Dicem early in my career, and I helped to be one of the ones who led bringing it into Amgen, the company I was with at the time. And since that time, I've recommended it on audits whenever I see the traditional sticky mats, the tacky mats, like there's a picture of there. And why? It's not just because it kind of looks neat and it's a newer technology, but also there's good data to back it up. There was an early study, which is, I think, one of the ones that really influenced me to help push for this, was a particulate control study that somebody from GSK had done back in 2002. And they showed that this technology, it's basically an electrostatic type interaction with particulates, had a mean percentage reduction of significantly higher than peel-off tacky mats. And recently I've seen another study that was done specifically on fungal contamination, which showed also the effectiveness of these tools. There's certain considerations with these tools. You have to have them large enough so that they catch multiple footsteps or wheel rotations, so that they have a chance to interact with the surface as much as possible before the person enters the clean room. And they also have to be cleaned regularly, depending on use. I don't know, Sarah, if you want to add anything more about Dicem to that. Okay, I guess I'll go on. So the next slide. So anyway, just just to wrap that thought up. So about DICEM, that particulate control study and particulate control is really powerful from these. So I'm no salesman, but if you still use tacky nuts, definitely recommend that you take a good look at those products, and you can have a sample sent out to you, or you can go to a facility where they have them installed in your area. So that's part of guarding against the human element. So people are the biggest source of contamination. Even when fully gowned and motionless in a clean room, an adult can generate up to 100,000 particles per minute. And those are particles greater than half a micron. And if people are being active, walking or things like that, it can potentially reach over 1 million particles per minute off of people. So about 10% of those particles carry microorganisms. And so gowning is really key. Cleanliness is important, personal hygiene. Training is so important for this. And proper aseptic technique, proper gowning, these are things that need to be trained, and you need to qualify your operators for. Again, I'm not going to get into detail on that, but there are some basic expectations depending on the classification of your type of operation that you're doing. The training needs to be very rigorous. I would recommend that for those who deal with these types of things and are concerned about training, there's some really innovative tools. One of the companies I work with is Cymorg for artificial intelligence empowered training and that can really help to keep people fresh, and it's a lot better than the read and understand training that we're so used to in our industry. One of the really key principles, though, for this is getting the human out of the loop. Wherever you can reduce the exposure of people to your process is the best practice. The next step, of course, for this is isolators with robotic systems for fill finish. That's where the industry is going. Definitely encourage you, if you're considering building a new fill finish facility, that you consider that. It may be more expensive up front, will be, but in the long run, it will definitely pay for itself. And I think it's only a matter of time before that becomes kind of the industry standard practice to get the human completely out of the loop for aseptic operations. So equipment cleaning is obviously key. Equipment needs to have smooth, non-reactive surfaces, minimal dead legs for traditional stainless steel type equipment, and needs to be able to disassembled fully for cleaning and inspection as much as possible. You have to validate all of those processes for cleaning, sterilization, and depyrogenation. Although all of these cycles are required, you want to make sure that they're thorough and robust and will work over a long period of time. Sometimes issues happen with systems over time, such as CIP spray balls getting clogged with debris, and then they don't work for cleaning the system. And so you have to ensure that you have controls in place on a routine basis to ensure that your equipment is still getting cleaned and sterilized appropriately. After cleaning and sterilization, you need to ensure that equipment is stored properly. Don't want to store it wet, for instance. It's a very common problem and can lead to significant contamination risks. Supply chain, obviously very important. You ultimately, the company whose name is on the product is responsible for the quality of that product. You're the ones that will be held accountable for it. And so if you are purchasing supplies, you need to ensure that you've done appropriate, rigorous supplier assessments, audits, to ensure that the quality of those materials. Recently, I just posted about it earlier this week. There was an FDA inspection at an API facility in India where they found bats and birds and rodents and cats in a warehouse, a GMP warehouse where API were being stored. That is having a dramatic impact on the industry right now, and I expect that will become part of that snowball of attention from the FDA, particularly on foreign suppliers and the importance of surprise inspections for foreign suppliers, which has been something that current FDA commissioner has been very focused on. So that there's many other controls listed here on how to ensure that you minimize the risk there. Whenever you have problems, you want to learn from those problems and investigate them thoroughly, and then make sure that you apply the lessons learned broadly to your organization and to your process. Very important in investigation to go to the scene of the crime. You can't do a good contamination investigation from a conference room. You really need to get the team out there, see what happened, look at the equipment. So often that's been the difference between, oh, it could have been this or it could have been that, or, ooh, that's not right. A lot of times when you're doing these investigations, you'll find multiple things that could have been the root cause. That's great. Fix them all. Sometimes you may not find the smoking gun. You may find a bunch of things that could have contributed, or maybe, you know, higher risk or lower risk. That's okay. As long as you find things, you dig as deep as you can to get to that root cause, and implement the corrective actions, and think broadly. If this happened in this system, could it happen in another system? If a problem happened in cell culture, could that same problem happen in manufacturing or in purification, for instance? That's what the regulatory agencies will expect when they review these investigation reports, which they will. Those are obviously a hot topic for them whenever they come to inspect. One of the great topics that's happening right now in this field is rapid microbial methods. One of the things that I sometimes get on my high horse about is the fact that we still to this day use the plating methods that were developed in the 1880s, where we're counting colonies on agar gels. And many facilities still use the technology. There are better technologies out there. They have been accepted by the agencies. Definitely encourage any company to investigate the use of rapid methods. They can save you up front. And oftentimes a good rapid method could pay for itself in a couple of batches saved. So it's very important to keep that in mind. This slide talks a little bit about bioburden in a water for injection loop, and how you could detect that and then potentially prevent contaminated water from being used for making a batch. So people always ask, well, what about inspection? Well, I said at the beginning, if you're doing things the right way, then you will be successful with inspections. But what the inspectors are looking for are really these things. They want to see that your design, you have quality by design implemented. So in this case, it's facility and process design. You can't use good results to justify poor design. Again, this comes back to that. You cannot test quality in. You want to do this quality risk management. That's that basic expectation. But you don't use quality risk management to justify poor practices. You want to use the right tools for those risk managements. Contamination control strategy begins with that detailed process mapping and a strong understanding of the process and the facility. And controlling your raw materials is so important. Ultimately, it's on you. So that's it. For legacy sites, older sites, you need to continuously assess your risk and plan for facility upgrades as necessary. So for those that are concerned about the regulations, make sure you review and align with the recent revisions for Annex 1 and evaluate those recent FDA actions. For example, that Novo Bloomington site 483 and OAI that recently was published. So summarizing, the preventing contamination requires that multi-layered defense. You have to have process understanding, risk assessment is foundational, learn from your issues, and practice continuous improvement. And ultimately, this leads to patient safety, operational success, and success with the inspectors. So I have a slide in here about regulation references, but we will jump right to my spot the problem game. We'll try that. And then at the end, we'll have a few minutes for questions. So the next few slides, there's 10, have some issues or potential issues that have been seen out there in manufacturing facilities. And your goal is to kind of see if you can spot the problem. You can type your answers into the Q&A field if you'd like, and that would that'd be good. Or you can just think about them. I'm not going to call anybody out by name or anything. So, but that's the way this works. The first one is hoses seen on a transfer panel. So, what might be wrong in this picture? What could lead to contamination issues? So then I'll go to the next one. Here's some operators in a clean room. This is from one of my recent LinkedIn posts. I use a lot of AI-generated art, and AI-generated art sometimes doesn't fully understand clean room operations. So think about what might be wrong in this picture. And then the next one is an operator performing an aseptic operation in a laminar flow hood. What might be wrong here? Maybe more than one thing. And the next one, this is a real photo from a facility I will not name, seen in a clean manufacturing area. There's some pans down on the floor underneath a piece of equipment. What might be wrong there? What questions might this trigger with you? Great. I love all these, all the comments and participation. It's great. Here is a picture of a clean area. There's a couple of issues here. See if you guys can spot those. But the cart is placed in its 5S location. See the tape on the floor that shows where the cart's supposed to be per their 5S program. What might be wrong with that and anything else that you see that jumps out at you on this cart? And last one, fairly obvious. We've got a wheel on a clean room cart. This is a real photo as well. Will not name the facility. Here's gowns in a gowning area. Again, a real facility. This one's a little bit for my SIP experts. Fittings on the top of a steam sterilized bioreactor. What might be wrong with this picture? And here's one for DICEM. Here's a traditional tacky mat. What does this tacky mat show you by looking at it? And I think this is my last picture, a box and a pallet in a clean room. What's wrong with this picture? Great. So I will just I'll go backwards through these and just give kind of my perspective on each one. There may be things that I'm not talking about, but I appreciate some of the comments here are great, really great. Every time you show something like this, you see multiple things, and which is good. That's the idea. Whenever you walk into one of these facilities, you want to think about that as you go. So going from the back one to the front and going very quickly because we are down to 15 minutes, I will talk about these. So the first problem here is that wooden pallet. Never want to bring wood into this type of facility. If you have to use a pallet, it should be a sanitized plastic pallet. The second big issue here is a corrugated cardboard box. Again, you never want to bring corrugated cardboard into a facility. So cardboard and wooden pallets can have pests. They can have mold, bacterial colonies growing on them. They can be very dirty, and it's hard to see the dirt or clean it off. Porous surfaces like that. Wooden pallets also can have chemical treatment that can cause problems for your facility. It actually led to a recall of medicine a number of years ago, chemically treated pallets. And so those are all things to think about there. The tacky mat, what jumps out at me is if you look at this picture, the cart was not clean. The cart that wheeled across this was not clean at the end of that tacky mat. So there's still dirt on those wheels. And so it's obvious that this tacky mat didn't fully clean, and that dirt was wheeled right into the facility. Let's see, previous one, the bioreactor. This is a little bit trickier, but this, if you look on the right-hand side of the picture, there's a pipe that goes up and ends in an elbow, and there's just a dead end up there. That location will never get steam sterilized in an SIP cycle. So that is a serious flaw with the design of this bioreactor. This is not entirely a real picture. It was a real picture of a bioreactor. I doctored it a little bit to make the flaw more obvious. But it's pipe sloping and designed for air removal and condensation removal, so critical for steam sterilized equipment. Gowns and gowning areas, it's pretty obvious. These are pretty dirty gowns. Again, this is a real facility. I was rather stunned by this one. Wheels on those clean room carts, rust obviously, can provide a place for microbes to hang out and grow, and can't be cleaned properly. So it should be removed and fixed and replaced. The 5S cart. So this cart has a couple of issues. First, there's a corrugated cardboard box there, and then secondly, it's placed right in front of the return for the HVAC system. So this is blocking the return, so potentially restricting airflow. The other thing is to think about is with many of these systems, the return feeds right back into the HVAC and goes through the HEPA filters and back into the room. And so any chemicals that are pulled into the returns, they'll pass right through that HEPA filter. And so it can be a source for chemical contamination wherever you have recirculating systems. Next one is fairly obvious. I think if they've got little pans under there, it's probably water dripping. And sure enough, each one of these pans was full of water. And where there's water, there will be mold and bacteria growing. Number of things. I saw a lot of comments on this one, and so this is an AI generated picture. Thank goodness. But the operator's got his hands right over the open operation there. Their pen and paper is inside the LFU, and the barrier is way too high. Those are just a few of the things that I noticed on this one. Clean room operators. This one I posted on LinkedIn, and a lot of people pointed out that the old guy on the right there his hairnet's not covering his hair properly, and his mask is not over his nose. So two major issues with this one that were pointed out. And then this one here is a classic. Hoses need to be stored in a way that they drain all the way. Even if a hose is dry after use, if it's hot, it may get moisture in it over time as it cools off from condensation. So you always have to store hoses in a way that they're dry. You can't entirely see the whole hose here, but there could be a pool of water here in the bottom of this hose that is not getting dried and just sitting there, growing contamination. So I think that's my last slide. So with that, I will turn it over to the Q&A.
Thank you very much for this presentation. And just a reminder for the audience, to ask questions, you can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they'll be answered at a later date. But we've got several questions already. Our first question: how often should the Contamination Control Strategy, CCS, and its underlying risk assessments be formally reviewed and updated?
So that's a great question. The general guidance here is at least annually, but it also should be evaluated whenever you have any significant changes to your process or updates to procedures, or if there's a significant issue that may require you to revise that contamination control strategy. Thank you.
Thank you so much. And our next question: what are the most common deficiencies cited by regulatory bodies like FDA 483s or EMA observations related to contamination control? And how can they be proactively addressed?
Yeah, so that's really the crux of it. The most common deficiencies recently have been, inadequate contamination investigation and CAPA, and a lack of understanding of the controls that are required to prevent that type of contamination from happening. How that can be prevented? Well, that's what this is about. Ensuring that you go back to the basics of how your facility and process is designed to control and prevent contamination, having those risk assessments in place, having a comprehensive contamination control strategy, and also ensuring that you're learning from issues, not just your own issues, but issues that occur, similar issues that may occur at other sites. I mentioned it before, but just to reiterate, whenever you see something in the news about some company getting either hit by an inspection or they have a major contamination event and they have to do a product recall or something of that nature, don't just think, too bad for them. Think, I better make sure that's not me, because that's really the good lesson there. And so paying attention to those things, learning from not only your own issues, but other issues, is a best practice for that. Next question.
That was a great answer. So this next question comes from Philip, saying, could the contamination control strategy be a part of the HACCP plan? A combination, since both CCS and HACCP use FMEA approach.
Yeah, so the HACCP is Hazard Analysis and Critical Control Strategy, and that's really kind of originated from the food industry, but it's a great model, and so you can use those same tactics to approach this, and so but yeah, that's a great approach. It really is a good tool. So
great. This next question says, can you expand on or provide examples for not confusing failure modes with root causes?
I'm sorry, what was the question again?
Could you provide examples for not confusing failure modes with root causes?
Oh, yes. So the failure modes, let me think about this. So a failure mode is a potential contributor. It's a mechanism which may lead to a contamination. A root cause is a more specific example of that. So, for instance, an improperly designed a bent pipe in a bioreactor, for instance, that doesn't get properly steam sterilized. That is a root cause of an issue. The failure mode is broader, of failure to achieve sterility in the system. So hope that helps. And if you need more clarity on anything, my contact information is there. You can also connect with me on LinkedIn. So
this next question says, Why is it difficult to detect toxins before media enters the bioreactor? Is there no measurement or instrument that can measure this in line?
So there are now. So for years, the limitation here was for microbial counting, you had to collect a sample, plate it out, and then wait, minimum of 3 days before you could get any kind of count. And typically 7 to 14 days before you had a good count of how many bacteria were present. So by that time, the batch is completely done. And so there was no detectability on that step. In the food industry, people often talk about, well, the basic rules of food safety. You have to keep hot food hot, cold food cold. There's very strict time limits on that and temperature limits on that. We all know that. When you go to a buffet and you see that they're not keeping things hot, you should start getting a little bit nervous about food poisoning. But for some reason in the pharma industry, I've seen that oftentimes operations kind of forget that. They forget those basic principles. And so you have a non-sterile or non-bioburden controlled process step, and people think, oh, I've got a sterilizing filter or sterilizing grade filter right here, so I don't need to worry about what's happening in this tank. It's because the bacteria will all be filtered out. The bacteria will be filtered out, but that's not the only potential cause of problems. I suspect that for biologics, some of our problems with stability that people have seen over longer periods of time may be related to protease activity from microbial contamination further upstream in the process. Protease and protein digesting enzymes that may slip through the cracks and not be fully detectable by our analytical assays. I also have a fear that for early clinical programs, we may sometimes be discarding a good product that because of failure in toxicology testing with animals, and those failures were not due to the product itself, but due to how the product was made, something similar to what my example with that bioreactor cell death investigation. And that problem could be killing off wonderful products that could be transformative for people. And that's one of the reasons I've long been an advocate for applying at least GMP principles, if not full GMP, which is not required, to early preclinical manufacturing. So.
That was great, David. Our next question is if this webinar will be available after. And yes, this webinar will be up on the Dyson YouTube page if you would like to rewatch any parts of this or revisit any of the questions asked from David today. Our next question says, do companies still execute gowning qualifications for operators?
Yes, they do. It depends on the classification and the grade and the type of operation that you're doing. It's absolutely a requirement for aseptic processing that operators are qualified and tested on a regular basis to ensure that they are meeting those requirements.
And our last question is, what were the most glaring deficiencies regarding the Novo Nordisk story earlier in your presentation?
Well, I think the one that hit everybody was the cat hair. This became the cat hair and blue spots 483, and it's really had a big impact across the industry, at least for those of us in quality and contamination response. But that was kind of it. And it really came down to repeated multiple incidents of contamination that were not thoroughly investigated or mitigated. This is not the first 483 from that particular facility. There have been multiple ones. And so they definitely have a lot of work to do, to solve this and get ahead of it. I really hope they do. We're all rooting for them. But in the meantime, again, these facilities, the problems that they have are not unique. I do a lot of audits. I've seen similar problems at many facilities around the world. So it's not unique, and I think the FDA is going to be really cracking down on this globally. I would expect when the government reopens that they will be out there with a vengeance. The current FDA commissioner has said he envisions returning to a time when the FDA sent out hundreds of letters a year for deficiencies. So they are definitely going to be gearing up for a focus in this area. So.
That's great. Yeah, I think we're all wishing Novo Nordisk the best. But if you're from the Novo team and you want David to come out and do an audit and help provide his guidance, or if you're interested in getting in contact with David, he is such an amazing expert and so knowledgeable in this field. His contact information is up on the screen right now. Thank you, David, so much for this amazing presentation, and everyone have a lovely rest of your day.
Thank you.